So our last speaker for the track today, before the plenary panel, is Jordi Riba. Jordi's going to talk about new findings from ayahuasca research from enhancing mindfulness abilities to promoting neurogenesis. Jordi Riba holds a PhD in pharmacology. He leads the Human Neuropsychopharmacology Research Group at Sant Pau Hospital in Barcelona. He has a broad interest in psychoactive drugs with publications on psychedelics, psychostimulants, cannabinoids, and kappa receptor agonists. He's been studying ayahuasca for over 15 years and has published nearly 40 journal articles and book chapters on the subject. He's also collaborated in the first clinical studies involving ayahuasca administration to patients with depression. His current research deals with the post-acute psychedelic afterglow and the use of ayahuasca in the treatment of various psychiatric conditions and the neuroprotective and neurogenic potential of ayahuasca alkaloids. Jordi.
Hi there, can you hear me? Yeah, first of all, I'd like to thank the organizers for having me once more. The last time was in 2013. If I remember correctly. So I hope I can show you some new data from our lab. And yeah, I hope you enjoy the presentation. So yeah, I've been in the ayahuasca research business for around 20 years. Most of it has happened at this institution, St. Paul's Hospital in Barcelona, and more recently I've started a collaboration with the Beckley Foundation. We have what it's called the Beckley Sant Pau Research Program, and many of the results I'm going to show you today are the result or the fruit of this exciting collaboration. So when I started research on ayahuasca like 20 years ago, I think I could safely say that I was studying a obscure psychoactive substance from the Amazon. But in these intervening 20 years, this is no longer the case, and ayahuasca has gone global. And now you can even find it in the general media. So I'm going to show you a few examples here. For instance, this article that appeared in the New York Times, it said ayahuasca, a strong cup of tea. So regardless of the content of the article, what I found fascinating was the section in which it was published. It was in fashion and style. It was not in, I don't know, drug abuse or health risks or anything of this sort. Recently, also the LA Times published an article on ayahuasca. Of course, there was the obligatory exotic picture of an ayahuasca session, and I found the title amazing. It said, Yahe tourism. Yahe is the term that's used in Colombia for ayahuasca. It said vomiting and visions in Colombia, comma, then peace. So things are really evolving, and now even Hollywood knows about ayahuasca. Here you can see Ben Stiller and Naomi Watts peacefully discussing an ayahuasca session and the life-changing experiences they've gone through. So I think that things, at least in the US, are really changing. So if you could please play for me this video. I could not resist the temptation of getting some aid from these Hollywood luminaries to tell you what ayahuasca is all about. So please go ahead with the video.
Yeah, I know. It's just we've got this ayahuasca ceremony this weekend with Jamie and Darby.
What's an ayahuasca ceremony?
I guess there's a shaman, and you have to wear white clothes, and you drink this sludgy liquid that is made from Peruvian root, and you hallucinate and vomit up your demons.
Okay.
So I don't think I could have summarized this better myself. So here are the first take-home messages from Naomi. Ayahuasca is a sludgy liquid. This is agreed. It's made from Peruvian root. The plant actually grows throughout the Amazon, and maybe the bark is preferred to the root in the preparation. But yeah, then you have this point that you hallucinate. I'm not sure about this, but based on the art that ayahuasca has generated, it certainly makes you see some funny stuff, right? And Then you have this vomiting aspect, and ayahuasca certainly works as advertised. And the only really true point of dissent with all these statements by Naomi Watts is wearing white clothes. I think this is actually optional, and at least based on our lab studies involving freeze-dried ayahuasca, if you're wearing your favorite black t-shirt, this will work just as well. So this is the basic introduction to ayahuasca. Now you know what this is all about, and I'm going to tell you about a little bit about the active principles that you found in this Peruvian root that she's mentioning. So the main ingredient in Banisteriopsis in the ayahuasca brews is Banisteriopsis caapi. This vine, this Amazonian vine, contains these alkaloids, the Harmon, Harmelin, tetrahydroharmine, and traditionally they've been considered to play just a secondary role in the overall effects of ayahuasca on the brain. So Harmon and Harmelin are potent monoamine oxidase inhibitors. The tetrahydroharmine is a weak monoamine oxidase inhibitor, and it also acts by inhibiting the reuptake of serotonin. So it was thought for a long time that the only role that these alkaloids played when you ingested ayahuasca was to make DMT, the next compound I'll be talking about, orally active. But recent studies have shown that perhaps the overall effects of ayahuasca are also modulated by these substances, because some in vitro findings show that they can interact at the cellular level with neural stem cells in the central nervous system. So this I'll show later on in my presentation. So why has so little attention been paid to these compounds? And the truth is that there are many versions of ayahuasca where only Banisteriopsis caapi is used, and then you can add to this main brew up to 100 different admixture plants. So most people who approach ayahuasca are on their vision quest, and they want to have this ingredient also in the brew, Psychotherapy, the leaves of this plant. And the leaves of this plant contain dimethyltryptamine, DMT, and this is a potent psychedelic, It's a potent psychedelic, so it's a 5-HT2A and 5-HT1A receptor agonist. It has other interactions with brain receptors, and more recently has been also shown to interact with the sigma-1 receptor agonist. So if you get this ingredient in ayahuasca, you will surely get lots of visions out of your money. So this has become very popular, and most people view ayahuasca merely as an oral form of DMT, but I think there's more to it than just this limited vision of the ayahuasca brew. So in this slide, I'm going to summarize how ayahuasca interacts with your brain. We've conducted many studies in the laboratory to find this out, and it basically modifies acutely brain dynamics. So here you have a summary of neuroimaging and neurophysiological findings. What we've seen is that it increases blood flow in frontal brain regions in the anterior cingulate cortex, in the subgenual anterior cingulate cortex, also in the medial temporal lobe, in the hippocampus, in the amygdala. This was measured using SPECT, that's a nuclear medicine technique. If you measure spontaneous electrical brain oscillations, you can find brain regions that are responsible for changes in power in these electrical oscillations. You can calculate what's called current density, and you see that the maximum decreases in current density occur in posterior brain areas, like the medial parietal cortex, a key hub of the default mode network, and also in visual regions. We have also measured what's called information transfer. This is acutely modified under the effects of ayahuasca. And recent studies by Draulio de Araújo in Brazil have shown that you do get increased bold response in visual areas. So until now, no one had demonstrated this, that you actually get activation of visual areas in the brain. He published this paper a couple of years ago showing these results. So basically what you have here is ayahuasca interacting with what we call multimodal brain hubs in the brain, the anterior cingulate cortex, the posterior cingulate cortex, the medial temporal lobe. All these regions are involved in key processes in psychological functioning, like emotion, memory, sense of self, like in the default mode network. And also you get activation in certain sensory areas like the visual cortex. So during the acute effects of ayahuasca, the neural hierarchies are transiently altered. What does this mean? This means that you can have a person with their eyes closed, and you can measure how at different leads how spontaneous brain electrical activity changes over time. And what you see is that there is an increase in predictability of signals in frontal brain regions if you take into account activity in posterior brain regions with a time lag. So that means that there's an increase in posterior to anterior information transfer. And this would not be surprising if someone were with eyes open. Okay, you get information, visual information, it goes to your visual cortex, and then it climbs up the hierarchy, this ladder in the brain. It goes to association areas, to areas involving cognitive control. But here you have persons that keep their eyes closed, and you are seeing this flow of information from bottom up and the decrease of top-down control. So sensory selective brain areas take the lead over frontal regions involved in executive function, in cognitive control. So this is what it means when I say that neural hierarchies are transiently altered during the effects of ayahuasca. We've also seen that these modifications in brain dynamics can lead to different patterns of thought. It facilitates what we call divergent thinking. This is a measure of creativity, and we recently published this study in which people were shown certain items, and they had to describe how these items were related in this task. So before we did ayahuasca intake, so their responses were the most conventional ones. So under the effects of ayahuasca, they came up with new associations, new ways of relating information that made sense as well. It was not nonsensical. It made sense, but it was not the standard way in which you would make these associations. So here's my question to you. Could psychedelics be facilitators of new ideas if they promote these increased associations in the brain, this new way of thinking? Well, there are some people who were successful in their careers, and they say that taking psychedelics had an influence in the creative aspects of their work. For instance, we can mention Steve Jobs, Francis Crick, who discovered the structure of DNA, or Carey Mullis, another Nobel laureate, who developed the polymerase chain reaction. So all these people think that psychedelics helped them come up with new ideas that were perhaps available to anyone, but no one had thought about them. Like the PCR was really a simple idea. It was just that you had to repeat a number of times a reaction that was already known, and that allowed that with tiny amounts of DNA, you could amplify that 1,000-fold, 1,000,000-fold, and conduct a series of analyses that were unthinkable some years before. So more recently, again, in my hospital, some people, perhaps with certain inclinations to or proneness to divergent thinking, came to my office and told me that they were working with mindfulness psychotherapy, and they could see some analogies between what I had described the ayahuasca experience was and mindfulness meditation. And they said, okay, could we measure this? We have some standard questionnaires that we administer to our patients. There are certain deficits in mindfulness facets that they found in many psychiatric conditions. They tried to stimulate these mindfulness facets in their patients, but this was a very lengthy and painstaking process. So they were curious about whether ayahuasca was able to induce this. So what we did was we set up a study, and we administered their standard questionnaires, the 5 facets mindfulness questionnaire, and the experiences questionnaire, and we administered these questionnaires before and after ayahuasca intake. It was just a single dose was involved here. So when we did find some some increases, some significant, statistically significant increases after a single dose of ayahuasca in some subscales of this questionnaire, of the FFMQ. That was in what's called non-reacting. In non-judging, people become less judgmental about their patterns of thought, about their lives, about what they think is wrong with their I don't know, life choices or events that happen to them. And also this experiences questionnaire measures what's called decentering. That means being more detached from your own thoughts, like viewing them that's something that just happens, but after a while it disappears. You don't have to identify yourself with constantly with some negative thoughts. So we found these interesting increases after a single dose of ayahuasca. Also, they had developed this so-called mindfulness or mind sensory composite index that is especially sensitive to people who have undergone very intense meditation practice. And the scores we obtained, the post-ayahuasca scores, were in the range associated with extensive meditation practice. So they were really impressed by this, and we have continued with this collaboration. So we tried to look for the neural basis underlying these changes in psychological facets that they try to stimulate in their patients. So we did an MRI study, and we analyzed functional connectivity. So these are the results of the post-ayahuasca versus the pre-ayahuasca connectivity. This is a seed to voxel approach. So we placed a voxel in the anterior cingulate cortex, and what we saw was that the anterior cingulate cortex increased its connectivity with the PCC, that area, that key hub of the default mode network, but also with the medial temporal lobe, an area where the key structures are the amygdala and the hippocampus that are associated with memory and emotion. So again, we find that what these increases in connectivity match what people report they experience under the effect of ayahuasca. And what was interesting here is that we took these measures from the whole sample, from each subject, this degree of increase in connectivity between the ACC and the MTL, and we tried to correlate this with the changes in mindfulness facets, and we did find a very interesting correlation. For instance, in this subscale, the non-judging subscale correlates with this increased connectivity between the ACC and the MTL. So we established this working hypothesis that maybe ayahuasca could be like a catalyst or a facilitator of emotional reprocessing. And we thought that perhaps we could include ayahuasca sessions within the traditional mindfulness psychotherapy these people in the psychiatry department are using with their patients. So the idea would be to try to speed up the process and try to boost the outcome of this mindfulness psychotherapy intervention. So we are working on that currently. So we already have some evidence that ayahuasca works in psychiatric patients. Some studies have been conducted in mood disorders. I have the honor to collaborate with 2 studies that have been conducted in Brazil. The first study, in the first study, we administered a single dose of ayahuasca to treatment-resistant depressive patients, and this was a small sample, was only 6 volunteers, or 6 patients, sorry, and we found very rapid and long-lasting decreases in ratings of depressive symptoms. So the improvements were already noticed 24 hours after administration of a single ayahuasca dose and were maintained up to 21 days, like 3 weeks. So we replicated this later in a larger sample in 16 patients. Again, the same results, the same impressive results. And more recent, Dr. Traulio de Araújo, also from Brazil, has conducted the first clinical trial in depressive patients involving a placebo-controlled, double-blind, randomized design. So he's published his results as a preprint, and he has presented this this morning. And he has shown that although there's a placebo effect, an initial decrease in scores in those patients who received the placebo ayahuasca, these patients rapidly went again to experience very intense depressive symptoms, whereas the other group who had received ayahuasca remained symptom-free for a longer time. So in this study, for the first time, someone has demonstrated that a psychedelic works better than a placebo in this context, in the context of treating depression. So we already know that ayahuasca can work to treat at least depression. So now I'm going to move on to these new findings we have on the beta-carbolines, the poor relatives in the ayahuasca brew, even though the Banisteriopsis piscapi is the necessary and sufficient ingredient to get ayahuasca. So we've studied the beta-carbolines in certain models, and what we were interested in was neurogenesis. So for a long time it was thought that new neurons are only generated in the embryo and perhaps in the early stages of infancy, but once you reach adulthood, you don't generate new neurons in the brain. So this is not the case. At least there are two areas in the brain where new neurons are being generated in mammals. At least it's true. It's true. They are being generated at a slow rate. But there's increasing research looking for molecules that might increase this ratio of generation of new neurons. So in order to get neurogenesis, you must prove, if you're testing a molecule, that you're able to induce these three effects. The first effect is what's called proliferation. That means multiplication of neural stem cells. Migration. This means that these neural stem cells can travel to integrate themselves into new brain circuits. And differentiation. That means that the neural stem cells can transform into other central nervous system cells, for instance, neurons themselves. So the hippocampus is one of these two areas in the brain where you have neurogenesis spontaneously, physiologically, and where this area is the target of the study of new molecules trying to boost this birth of new neurons in the brain. So here in the slide, what you can see is the small bluish cells. I'm not sure if you can see this here in the slide. Or sorry, the green cells are the neuroblasts. These neuroblasts migrate. They develop into granular cells, and they integrate themselves into circuits of the hippocampus. So the hippocampus is an area that's in the brain, the medial temporal lobe, that's basically implicated in many forms of memory, and it's one of the first areas that gets affected in diseases such as Alzheimer's disease. So another researcher in Brazil, so we decided to conduct studies on neurogenesis, but another researcher in Brazil I didn't know of also conducted a similar study. So he was able to demonstrate that Harmon stimulates proliferation of neural stem cells, and he has published this paper in PRG. It has appeared recently, so he already demonstrated this. And now I'm going to show you the results of our own research. This has been done in collaboration with a research center in Madrid. They belong to an organization called the Spanish National Research Council. And what we've done is tested several beta-carbolines in this neurogenesis model. I'm going to show you results for Harmon. So the first step that the first results I'm going to show you here deal with proliferation. So here what we are measuring is what we call the number of neurospheres and the size of these neurospheres. What we do is we extract neural stem cells from the brains of rodents, of mice. So we culture those neural stem cells in a medium, and they form a sphere. This is what's called a neurosphere, and we can test different compounds in this medium and see if they increase the size and the number of these neurospheres. So we tested Harmon in this model, and the top panels here show the results for saline. The bottom panels show the results for Harmon. So first, if you culture neural stem cells under saline conditions, you see that certain neurospheres are formed. If you add Harmon to the medium, you see that the amount of neurospheres is much larger. And if you compare the size of the neurospheres, you must remember that this is three-dimensional. This is a sphere, is a volume, right? The size is much larger under Harmon. That means that each neurosphere has a much larger number of cells. So this is from the subventricular zone of the brain. We also obtained neural stem cells from the subgranular zone of the hippocampus of these adult mice. And again, under saline conditions, you get some neurospheres being formed. Under Harmon, the number is dramatically larger. And finally, when we measured the size, the volume of these spheres, when you administer Harmon, so you get a much larger size. So what this slide shows you is that Harmon stimulates the creation of this neurosphere, it increases the number of cells that are present in these neurospheres. Could you please play the video on the left-hand side of the slide? So here what we are measuring is migration. So you have the neural stem cells, and you measure in time how far they move away from the original neurosphere. So this would be after saline, and if you can play the other video on the right-hand side, please. This would be after Harmon. So that you can compare this more easily, you can see the range that the individual neural stem cells reached after saline on the left-hand side, and the distance they reached after Harmon. So Harmon also stimulates this second aspect of neurogenesis. And finally, the third aspect that we measured was the capacity of Harmon to influence neural stem cell differentiation, in this case, into neurons. So here in this slide, the color code of the image is the following. So in blue, what you have here is a stain that shows you every single cell, because this stain stains the nuclei of the cell. So whether it's a stem cell or it's a neuron, they are going to stain every single eukaryotic cell you have in this culture. So after saline, this is all you get. Blue is just for the nuclei. The green staining shows you young neurons. The red staining would show you mature neurons. Here are no young neurons, no mature neurons, just a single greenish and red spots. If you repeat the same test on the Harmon, what you get are these dramatic effects. So Harmon, again, is stimulating this third step that comprises neurogenesis. So based on these interesting results, we must not forget that this is in vitro. We can ask ourselves what the potential areas of future research could be. We have already demonstrated that ayahuasca works for depression, and even if we include a placebo in the study design, we have anecdotal reports of people being able to give up excessive alcohol consumption or highly addictive drugs such as cocaine or heroin after participating or getting involved in the ayahuasca taking groups. So this is another area we're interested in working in. And based on that emotional reprocessing working hypothesis we have, we think that it could also work well to treat psychological trauma. And in fact, we have designed a study. We are trying to get underway, integrating this mindfulness psychotherapy and ayahuasca intake to treat psychological trauma. So these are the most immediate goals. What I see, it's more feasible, but based on the results I've shown you earlier, we could also, think more creatively and ask ourselves whether cognitive deficits associated with normal aging could respond to these compounds present in ayahuasca, such as memory loss. Also, neurodegenerative disorders. There are several animal models where these compounds could be tested. Animal models of Alzheimer's disease, Parkinson's disease, Huntington's disease. So I think these are some exciting new areas where our research should be moving to. So just to conclude, I want to mention this upcoming therapeutic study we have in Barcelona, in our institution at Sant Pau Hospital. And It's this idea of combining mindfulness psychotherapy and ayahuasca. The target population will be patients with psychological trauma. We actually have received governmental funding to conduct this. Unfortunately, the economy we are in has only allowed our stingy government to give us 1/4 of the total amount we need to get this underway. So we are thinking to start a crowdfunding initiative. So this is still not set up, but remembering the first time I landed on planet San Francisco 20 years ago, I was constantly being greeted by this message everywhere. Brother, can you spare a dime? So this is my question to you, billionaires in the audience and non-billionaires in the audience. If you think you can spare a dime, you think you can, you find this research interesting, you would like to support it somehow, you can write me to this email address, ayahuasca.crowdfunding@gmail.com, and I'll let you know when this fundraising initiative will start. And here concludes my presentation. Thank you for your attention, and I'm open to questions.
Why don't we open the floor up to questions? They're going to put a mic in the aisle for you, so we can line up.
Yeah, hello, thank you for your talk. It was very pertinent to hear the information about the possibility of this neuro research with ayahuasca expanding, I'm sorry, I don't know your technical terms, but helping to possibly prevent Alzheimer's, which is sort of the terror of everybody who's my age, who's seen anybody go that way. But I have a different question, and that is, I've heard today a lot about ayahuasca and other psychedelics being effective in addictions, such as to alcohol or to cocaine. I haven't heard anything about tobacco, cigarette smoking. Do you know of research, or have you done
Yes,
research?
the Johns Hopkins team has conducted at least one study with psilocybin for nicotine addiction, and I think they had very positive results.
Okay.
I think some of the members of the group are in the audience, so you should ask them.
Yeah, sure. Thank you.
No doubt you're familiar with the work on Harmelin, on ibogaine and heroin addiction, which was terminated after they discovered that there were prunings occurring in the cerebellar vermis and loss of Purkinje cells. And then there was a publication that showed, not only does ibogaine do that, but so does Harmelin. And the question might be, are we doing ourselves good by stopping heroin addiction in essence by ayahuasca? And is this not a good thing, and it should not have been stopped? And so you're showing increased neurogenesis. Could you look in the cerebellar vermis and see the effect of Harmelin and other beta-carbolines there? It would be very interesting to know.
Yeah, if I remember correctly, Harmelin was studied as well in this context, and it was also found to induce some negative effects on cerebellar cells. But Harmelin is the beta-carboline that's present in the lowest concentrations in ayahuasca. So
nevertheless,
yeah, but compared to Harmin or Harmelin, maybe it's there's 100 times less Harmelin than the other two. And of course, you could also think of using the pure compounds. Right. You could just forget about Harmelin and try to work with Harmin and tetrahydroharmin.
Actually valuable and that some of us are over, we have too many Purkinje cells in the vermis because of our stress, and that the reason that these are effective agents is because they're pruning them back down to a normal level.
So you view that as a positive result?
Yes.
Okay, that's an alternative.
Anyway,
That's
thank you very much.
divergent thinking, I would say.
So in the study that you discussed, neurogenesis, if I'm correct, the stem cells were taken outside of the brain and it was studied in isolation, right?
Yes.
Have you actually looked at this effect within a living model? Because I feel like within isolation, there's a lot that we can't really rely upon as far as the results.
Not yet, but we plan to conduct these studies.
Do you know of any others that are currently doing that?
I don't really know if someone else is doing this in vivo.
Okay, thank you.
Hey, thank you very much for your speech or your talk. So I was really interested about Harmelin or Harmine for neurogenesis because I remember reading a while back about the potential for Harmine to be an anti-cancer agent by attacking topoisomerase, which seems like it's sort of a contradictory kind of action with the neurogenesis. And both of these have been shown, I don't think in vivo, both have been shown in vitro. So I was wondering what conjecture you might make on the mechanism behind that.
I'm aware of these contradictory findings, but I don't have an answer to what the reason is. So the study that was conducted in Brazil with Harmon, they examined the mechanism of action under the neurogenic, the proliferation effects they were observing, and that did not depend on monoamine oxidase inhibition. It went through another pathway. So it might be that in some instances you're blocking a certain pathway and then you get these neurogenic effects. It might be that another pathway is enhanced in cancer cells and or depressed in cancer cells, and then you can get opposite results.
Right, like this may be a specific environment for cancer cells. Thank you.
Welcome.
Hello, thanks. Thanks for your research, and this is fantastic. It's CME lecture, and I was wondering if you could elaborate on question 2 that we have. Could you inform how you would advise patients on the long-term effects and consequences of ayahuasca based on the published clinical? What would your line be to a patient who told you they were ready to go to ceremony this weekend?
I would not advise people to go to ayahuasca ceremonies.
But they're going.
Yeah, they told me that getting into psychedelic drug research was a career killer. It hasn't killed me yet in 20 years, but I don't know, maybe if I start advising things like that, I might put myself at risk, you know. I think people should be careful. There might be interactions with other substances. The psychotropic effects you get out of ayahuasca are quite intense. It might not be for everyone, depending on your, I don't know, your vulnerabilities. Maybe people should refrain from experimenting with these drugs. All these studies we've conducted in humans were healthy volunteers, young healthy volunteers. They went through psychiatric interviews. They went through medical tests, blood analysis, all sorts of tests to make sure that they had no health problems. So when I report that no untoward effects were found in these studies, these are carefully selected individuals. So I cannot guarantee that some patient suffering from some condition won't suffer any adverse consequence from ayahuasca use. So I think people should be careful. And these results are completely preliminary. This is an in vitro model. This doesn't mean that it works for Alzheimer's or anything. There are many other small molecules that have worked in these kind of models, and later on they have proved that they have no efficacy in a clinical context. But nevertheless, we have to look into this. But this is my honest answer to you.
Thanks. I guess maybe just a little bit of elaboration on just the MAOI effects. Is that in the scope of this talk?
There's a lot of variability between the few studies that have been published. For instance, in our sample in Barcelona, most people didn't have any measurable levels of harmine in blood. So it got degraded very rapidly. What we could measure was harmol. That's the demethylated metabolite of harmine. But in studies conducted in Brazil, they found that people had high levels of harmine in their bloodstreams after taking ayahuasca. So there's individual differences there. So if you degrade harmine very rapidly, perhaps you're less at risk of getting interactions with other medications of certain foodstuffs. But if you're a slow metabolizer, maybe you can get dangerous interactions from it. If you combine it with, I don't know, phenylephrine or any other sort of stimulant of the sympathetic nervous system, or methylphenidate, or dextroamphetamine, or whatever. So it may change a lot from one person to the next. I would advise people to be careful, but because in principle, monoamine oxidase inhibitors have lots of interactions with other drugs.
Thank you.
You're welcome.
Hi, Jordi. I was wondering if you know about, or if you could hypothesize about the effects of varying amounts of tetrahydroharmine in a particular brew of ayahuasca, tetrahydroharmine being serotonin reuptake inhibiting, and does that affect the quality of the experience, or does it affect more or less DMT visions and so on?
In this same study where we were not able to measure plasma, significant plasma levels of harmine, we did measure large amounts of tetrahydroharmine. And tetrahydroharmine, or THH, has a longer elimination half-life than other compounds in ayahuasca. So it stays in the body for a longer time. So it may contribute to the midterm effects of ayahuasca. So I haven't shown this in my presentation, but we found that THH was also neurogenic in this model. So perhaps you get harmine completely degraded in first pass metabolism. It doesn't reach the brain in the significant amounts, but perhaps tetrahydroharmine does, and it exerts some effects there. So ideally, we should devise a study in which we administer tetrahydroharmine alone, see what the effects are, compare this with combinations with DMT, and the three compounds together, THH, harmine, DMT, etc. But of course, you need gasoline to get this running, and gasoline is money in this context. Yeah. So, and we are chronically underfunded. So I don't know of anyone who has tried this yet.
Could you hypothesize that because it's acting on the serotonin reuptake inhibition, could it also be acting on the other 5-HT2A receptor agonist, i.e. DMT? Could it be amplifying the DMT?
You're probably increasing your endogenous serotonin levels. But what the final effect this has on the effect of DMT on the 2A receptors, we can just speculate, because these increased levels of serotonin are going to interact with all serotonin receptors. So I really cannot tell you what the final outcome will be.
Okay, muchas gracias.
Hi, Jordi.
Hi.
So I've always seen the beta-carbolines to be active in the gut to help DMT make it to the brain through the bloodstream. But I've never thought of them as something which makes it into the bloodstream, let alone cross the blood-brain barrier itself and be active in the brain. And from the studies that I've read, so I saw your study, but there's been like 3 or 4 other studies which looked at ayahuasca and beta-carboline content in the blood, and there's no real consensus that it actually makes it past the liver into the blood, let alone into the brain. First of all, could you consolidate what you know about that, because you definitely know more than what I would, and which results are the most relevant?
Well, tetrahydroharmine is highly lipophilic.
Yeah.
So we've done some tests that show that it crosses the blood-brain barrier. And there's consensus in all studies that have assessed the pharmacokinetics of the active principles of ayahuasca have been able to measure THH. So I'm sure that some THH reaches the central nervous system. With regard to Harmon, in my study, I didn't see any significant levels in plasma. But in the study done by Jay Scalaway, Charles Grubb, etc., they were able to measure Harmon. And more recently, I think Eduardo Schemberg also published some data from a study in which they were also able to measure Harmon in plasma. So it may change from one study to another. I don't know. Maybe it has something to do with whether you are a slow or a fast metabolizer. Maybe the subjects we recruited for our study were fast metabolizers, and for this reason we didn't measure high Harmon levels or almost no Harmon at all in plasma.
And it's the same for Harmelin?
Harmelin, we could quantify it. Yeah.
Okay.
Yeah, there was no problem with that. We found the metabolite, but also measurable levels of Harmelin.
Okay. Thank you.
But these are low.
Yeah, it's still
compared
confusing.
to tetrahydroharmine levels.
Sweet. Thank you.
Welcome.
Thank you. My question is, would the ayahuasca, which contains only the wine, not the DMT component, be effective for this positive effect for neurogenesis?
I've been asking around to other colleagues who know more about anthropology and the use of the ayahuasca vine in a traditional context. They haven't been able to tell me whether there's groups that exclusively use the bark without any additives. So I don't know if it's currently being used by any human groups in this way. In principle, if you can get these results, these effects I showed you here from the beta-carbolines, perhaps you wouldn't need the DMT. So it would make sense to test these compounds now in vivo in animals, and if results are positive, maybe do some studies in healthy humans, and then perhaps move on to patients and see whether we see any positive changes. But that would mean that perhaps you don't need the DMT to get these effects. Of course, if you are on the vision quest, you will be disappointed, yeah.
Would it be easier to do this research with wine-only ayahuasca for legal reason? And would
definitely, I think it would be more easier to convince regulatory authorities that you want to use these compounds for medical purposes, and they are not listed in this Schedule 1 list of troublesome compounds.
Thank you.
Hi, Professor. Thanks for giving this talk and for the amazing work you do and all of this. Like, it's really exciting stuff. I'm actually had a professor who was really interested in mitochondrial health at one point, and I was wondering if these harmful alkaloids have been studied, or if anyone's thinking about doing studies, or if there's anything in the literature about MAOI's effects on mitochondria and their proliferation.
Nothing that I can remember now. Maybe there is, but I don't have the information here. I don't know.
Hi, my question was about the treatment studies for depression. I'm curious if you could talk a little bit about kind of what the setting was for the patient when they took the ayahuasca. Was it done in sort of kind of the context that they're doing some of the MDMA studies now and the psilocybin studies, or?
It was in a clinic, in a hospital. It was a hospital setting.
Did they have like therapist guides, or was it more sort of, just kind of let them do their own thing? I'm just curious about sort of from the psychotherapeutic perspective, what was going on.
Well, they try to prepare the room to make it as comfortable as possible within the hospital, and certain music was played during the sessions, and they tried not to interfere much with the experiences the participants were going through. So unless some help was needed at some point. But yeah, the approach was to let people go through the experience, supervise them, be as supportive as possible, but try not to interfere.
Thank you.
Hey there. So I was wondering if you know of any research that's done on ayahuasca in regards to ALS.
ALS?
It's Lou Gehrig's disease.
Amyotrophic lateral sclerosis?
Yep.
Okay. I'm not sure if someone's done this. But recently, a patient approached me in Barcelona asking if this could work for them. And, it's this pretty uncomfortable situations in which you don't know what to say. Because, these patients are desperate. They are ready to try anything, but we don't have the evidence to tell them that this will work. So.
Okay. Thank you very much.
For now, I could not say that this could have any beneficial effect on this disorder.
Well, thank you.
Hello, and thank you very much. How long were the antidepressant effects of the ayahuasca effective for the depressed population?
They were measured up to 3 weeks after the single dose administration. So, and then the study stopped there. So there was no follow-up. Any other questions? No? Okay, thanks for your attention.